The title for this podcast is Mendelian Genetics. It's found in Chapter 10 of your textbook. So in the latest activity that you did in class, you were looking at Mendels Peas. Mendel, remember, was an Austrian monk who part of his scientific experiments were growing pea plants and trying to figure out how they passed on their traits. Some of the traits he looked at were the color of the peas, the color of the flowers, how tall or short the pea plants were. And he cross-pollinated a bunch of different plants to see how those different traits were passed on. When he was all done, he counted up his data and came to some conclusions based on the numbers that he got. Number one, what Mendel figured out was that any time that he crossed two purebred plants, he got all of the same traits in the F1. All right, so we need to kind of pull this apart and figure out what exactly this means. So the first word we want to look at is purebred. So this word purebred here, okay, is something that you hear lots of times in like dog breeding talk and things like that. Purebred basically means that these are plants, in Mendel's case, that had parents that looked the same as them. Okay, so if you didn't write that down yet, please do that. Purebred, parents look same as offspring. Okay, so we take two plants whose parents look the same. So in other words, take two green pea plants whose parents were also green peas. And when he cross-pollinated them, he got all the same traits. So they looked the same, traits being, in this case, the green peas, in the F1. Now you saw this F1 when you did the foil peas in class. F1 stands for filial one. Filial is a Latin term for first generation. So the children, or the offspring of this cross, all look the same. And again, go back to the foil pea experiment you did. In your F1 cross, all of those peas that you found were smooth peas. Right? So we get all the same traits in the F1, all smooth peas when you cross two purebreds. But then the curious thing that happened, and the thing that he didn't expect, is that when he crossed those offspring, so those F1 offspring, and he cross-pollinated those, just like you did with your foil peas, he got different traits. Now, those offspring, those F1s that we had up here, are now called hybrids. So we cross hybrids. Hybrids have different traits. So not the same traits as the parents. And they resulted in different traits showing up in that first generation. So when you did your foil peas, and that second time you did that, it was actually the F2, you got some wrinkled ones starting to show up. And this surprised Mendel, because back in his day, he didn't think, and a lot of scientists in his day, didn't think that traits necessarily just could pop up out of nowhere. A lot of scientists in his day thought that blending was how traits were passed on. So if you had a tall parent and a short parent, all of the offspring should be medium, somewhere in the middle. That's the blending hypothesis. So the fact that they came up with different traits in his pea plants, so things that looked different from the parents, was very surprising. Okay, it wasn't like the parents, it wasn't in between the parents, it was completely different from the parents. So this surprised Mendel. Based on what he found, he made a couple hypotheses that he thought would explain why things were happening the way they were. So one of those hypotheses, this is what he discovered, number 2, he said that each of these traits must have two versions, and he called these versions alleles. So in his case, his versions were, again, in the pea plants, tall or short, smooth or wrinkled, green or yellow. So in his pea plants, he said that all of these different versions were called alleles, okay, the versions of the trait. Then he took it a step further, and he said in each case, one of these traits has to be dominant, and one of these traits has to be what he called recessive. So let's talk about what he meant by that. By dominant allele, or the dominant trait, what he meant is that this allele covers the recessive allele. And the recessive allele only shows when there's no dominant. Allele. So if we go back to Mendel's examples, let's take tall versus short pea plants. Okay, if the dominant allele is tall and the recessive allele is short, and every trait has two versions, okay, that means that every pea plant has two versions. If we get a pea plant that's tall as its two versions, that makes sense that it's going to look tall. But what Mendel said is if we have a pea plant that's got tall, short as its two versions, it's going to look tall. Because again, that dominant allele covers up the recessive allele, and all we see is the dominant allele in his pea plants. The other interesting thing that happens, besides the covering up, is remember, we can get that short allele to appear if we've got two short versions. So if a plant is short, it will look short. All right, so that was kind of the genius behind what Mendel figured out, was the idea that every trait, again, has two versions. Okay, we now call these versions alleles. He called them characters or something like that, but we are going to call them alleles in class. And depending on the different types of alleles that come together in a pea plant or in a human or in any other organism, it determines what we'll actually see as the trait. Again, two dominant alleles, and the trait looks dominant. One dominant and one recessive, and the trait looks dominant. Two recessive alleles, and the trait looks recessive. Right? Those were the kind of rules that he came up with. So, taking that into account, Mendel then went one step further. Now, Mendel didn't know anything about meiosis, because in the 1800s, they really didn't know about chromosomes and genes and DNA and things like that. They couldn't see cells going through meiosis and dividing to make these different gametes. But he did know that something had to happen when reproduction took place. Okay, because these alleles were showing up in some cases but not in others, he knew some things had to be happening when these cells got made. So he's got two laws that he proposed that explained how these alleles worked. His first law was called the law of segregation. And he said that during the formation of gametes, so when the pollen or the eggs were made, the alleles must have been separated from each other. So in other words, you can think of this like a deck of cards. Okay, the mother's alleles are one deck of cards, and the father's alleles are another deck of cards. And when gametes are made, so the pollen or the eggs in the case of pea plants, the alleles are separated. So they shuffle the decks and they all get mixed in together. So the alleles don't necessarily travel together. So for example, if the mother is a tall pea plant, those two tall alleles don't have to stay together when the egg is made. Instead, they can be separated apart, like shuffling a deck. The second law that he came up with was called the law of independent assortment. And this is when you take your deck and you deal it out. So again, not only are the alleles all shuffled, but then they're randomly distributed into those cells that are finally made in meiosis. Again, he didn't know anything about meiosis, but the numbers that he came up with in his pea plants told him that these alleles had to be randomly sorted out. In other words, randomly dealt out to those 4 gametes that are made in meiosis. So we're going to look at meiosis one more time and now apply Mendel's principles to this idea. So just to review, meiosis is the special form of cell division where the number of chromosomes are halved. It only occurs in reproductive tissue to make those reproductive cells, the egg, the sperm, the pollen, so that they each have half the DNA in them. We talked about there being 2 divisions in meiosis. Meiosis 1, where the homologous chromosomes lined up and then divided. Remember, the homologous chromosomes are one from mom and one from dad that are identical. They have the same genes on them, but different versions of the gene. In other words, now we know those versions are called alleles. The second division is when those sister chromatids line up and divide. And this is where we actually split the chromosome number in half and get those 4 gamete cells. So 4 sperm, egg, or pollen cells. So based on that, we're going to go back to Mendel's principles and see where in meiosis do these things actually occur. And just a reminder, Mendel didn't know anything about meiosis, but he still made these laws, and now meiosis explains these laws. So we're going to see how that works.